Hi, my name is Carlin, and I will be talking about the effects of lung volume on sympathetic transduction of blood pressure. This summer, I had the pleasure to work in Dr. Philip Millar's Cardiophysiology Lab alongside master's and PhD students. Our lab does a lot of work with microneurography, whereby an electrode is inserted in the peroneal nerve in the leg to measure nerve traffic. The resulting recording is what's seen here in blue, also known as a neurogram. This technique can be used to measure various types of nerve traffic, but our lab's focus is on muscle sympathetic nerve activity, aka MSNA. This is the outgoing signal from the central nervous system that innervates vascular smooth muscle, and it's important in controlling systemic blood pressure through regulation of peripheral resistance. We see in this diagram that following what is called an MSNA burst or burst cluster, there's a subsequent rise in blood pressure. And after a period of no bursts occurring, blood pressure starts to fall again. And then another MSNA burst occurs, leading to a rise in blood pressure. And so the cycle goes. Sympathetic transduction then is a way to quantify the changes in a variable such as blood pressure following an MSNA burst. For each variable of interest, the serial changes are calculated for 15 cardiac cycles or 15 heartbeats following a burst. For example, on the neurogram, we see that various bursts have been marked by a colored dot. If we look at this large burst marked by a blue dot, there's a blue arrow on the blood pressure tracing associated with it. And we can graph these changes as seen here on the left. On the other hand, we also want to see how the variable changes when there isn't a burst. So for every heartbeat not associated with the burst, termed no bursts, we also calculate the changes across 15 cardiac cycles. So if we look at this green dot above the ECG tracing and the associated green arrow below the blood pressure tracing, we see the changes in blood pressure graphed on the right. Studies have shown that MSNA is influenced by respiration, such that the greatest occurrence of bursts occurs during expiration and at low lung volumes. Additionally, previous research by our lab demonstrated that baroreflex sensitivity, which is related to sympathetic transduction, is greater at low lung volumes compared to high lung volumes, while there's no difference between inspiration and expiration. So we decided to investigate low and high lung volumes as it relates to transduction. We used previous data collected from 17 healthy male participants who underwent an 8 to 10 minute baseline of continuous measures of blood pressure, heart rate, MSNA, and respiratory variables. Our results showed that sympathetic transduction of diastolic blood pressure was larger during low lung volumes compared to high lung volumes. And this difference was found to be statistically significant. We also looked at transduction of heart rate, stroke volume, total vascular conductance, and cardiac output in order to determine the influences on this pressor response. First of all, we found that the pressor response was influenced by heart rate and stroke volume. At low lung volumes, we can see that heart rate increased, and at high lung volumes, heart rate decreased. On the other hand, we can see here that at low lung volumes, stroke volume went down, and at high lung volumes, stroke volume went up. Given that total vascular conductance in response to an MSNA burst was similar across volumes as seen here, the difference in the pressor response we saw is not likely attributed to altered vasoconstriction. However, since we know that blood pressure is changing and TVC stayed the same, that means that cardiac output is the likely culprit. And as we can see here, cardiac output was increased at low lung volumes compared to high lung volumes. In conclusion, we found that non-neural factors such as respiration play a role in sympathetic transduction of blood pressure through modulation of heart rate and stroke volume. It is important to note that these changes in heart rate and stroke volume can be regulated by various factors, either through neural interactions such as MSNA or through mechanical factors such as changes in venous return. Thank you for listening to my presentation.